Hello again, Biology 300 students. Mr. Park here, and we're going to continue on from segment number 1 of the Central Dogma onto segment number 2 of the Central Dogma, which will help continue on with your notes that was found in the yellow packet, or the packet that had the yellow cover sheet. Okay, so in the first one we talked about, what were the requirements of genetic material, what were some of the, why DNA was considered to be the genetic material versus proteins, and how the three different scientists, Griffith, Avery, McCarty, and McLeod, and Hershey and Chase, proved the idea that DNA was actually, in fact, the molecule that carried the genetic information. So now we're transitioning into the idea of what the actual structure of DNA is. Okay, DNA is abbreviated, you can see here, as it stands for deoxyribonucleic acid. Okay, so the abbreviation there is DNA, and it's classified as a nucleic acid. And again, nucleic acid is one of the large macromolecules of all living organisms. Okay, we talked about macromolecules before in organic chemistry, meaning your lipids, your carbohydrates, your proteins, and now your nucleic acids. Okay. Some characteristics of your DNA structure is it's long strands, double-stranded. The molecule is made up of monomers known as nucleotides. So these monomers, you put a bunch of monomers together, you create what we call a polymer. Well, in the case of the nucleic acids, we're talking about putting a bunch of nucleotides together that would create a polynucleotide chain, which basically is our DNA structure. Okay? And basically your DNA stores and transmits the hereditary information by coding for the production of proteins. So through those different experiments we had talked about in segment 1, the scientists proved that DNA actually carries this genetic information, and what it ultimately does is codes for the production of proteins, which we know occurs within the ribosomes, and those proteins will go off and tell your body really what to do. Okay, so basically out of here you need to know that DNA is a nucleotide or nucleic acid. The building blocks of nucleic acids are your nucleotides, and we know it stores the genetic information that will code for your proteins. Okay, so the basic unit of DNA is the nucleotide, as I stated before. There's 3 different parts, okay, with your nucleotide. As you can see here on the screen, the 3 different parts that you have is your phosphate group, your deoxyribose, or which we call the 5-carbon sugar. You have 1, 2, 3, 4, and 5, and then you have your nitrogen base. Okay, so there's your deoxyribose sugar, your phosphate group, and then also your nitrogen base. And by building these up and putting them together, we create our nucleic acid. And these structures are our monomer, or the building block of your nucleic acid. Okay, so now we're looking at, as we mentioned, those 3 different parts of nucleic acid. One of them, the nucleotide, one of them is the nitrogen bases. And your nitrogen bases is broken into 2 different classes, as you can see on the screen here. You have your pyrimidines and your purines. Okay, your pyrimidines, this is the way I remember which ones are which, is in the pyrimidines, you notice that the pyrimidines have a Y in it, and so does your thymine, and so does your cytosine. Okay, and then your purines, you have adenine and guanine. Okay, so your nitrogen bases, you have 4 nitrogen bases, and again, your adenine and guanine are your purines, your pyrimidines are your thymine and cytosine. Okay, and these help basically kind of put your double-stranded DNA together. Okay, so your nucleotides, as I mentioned earlier, your nucleotides are, again, your building blocks. And when we piece them together, we create this long chain, which we refer to as a polynucleotide chain. So you can see here, here's one of your nucleotides right here. Okay, it's a blown out picture of it. Or if you look at this area here, you can see here's your phosphates, your sugar, and then it's connected to your nitrogen base. And again, you have your phosphate, your sugar, and your nitrogen base. And when we group those or we bond those together, we create our polynucleotide chain, okay, which is one side of the double-stranded side of DNA, and then we have to we would connect those with hydrogen bonds to the second side to create the double strand. And we'll take a little bit closer look at that here in a second. Okay, so making these polynucleotide chain, okay, as I've mentioned a number of times, if you remember the terms and the prefixes that we have done before, poly means many, and then we have our nucleotide, which is the building block of your DNA. And you can see here what I refer to as your sugar phosphate backbone. Okay, you have your phosphate group and your sugar group. Phosphate group, sugar group, and they're linked together. Okay, and here's your nitrogen bases. Okay, your thymine, guanine, cytosine, and adenine. And depending on what order it is, and depends on what proteins eventually it will code for. Okay, it just all depends on the order. Kind of like the alphabet. You know, when you think about the alphabet, the way you put your letters together and the order of the letters basically tell you the words, same idea. When you put together your letters of your DNA, and the order they're in and the length depends on what proteins you'll eventually be coding for, and eventually you'll be able to tell what your body is going to be doing. Okay, so basically this makes up the sides of the ladder, as I mentioned. So if I ever ask you on a test or a quiz and I say, you know, what's the size of the ladder, or what's the backbone of the DNA, you should always tell me it's the sugar phosphate group. Okay. All right, so the double helix now. Looking at the double helix, this is basically the DNA consists of two polynucleotide chains. Okay, so you have two sides if you think about it, and the chains wound around each other to form what they refer to as the double helix. So if you unwind it, it looks like a ladder, but if you wind them back up, it looks like it's what we refer to as a double helix. Okay, and the famous person for this, the scientist for this, is Rosalind Franklin. Okay, she basically worked with X-ray crystallography, and she was taking pictures of DNA. And you can see here in this slide here, you can see this is the double helix that you can see the helical shape. And here obviously is Rosalind Franklin. And actually she had died prior to the fact of, I guess you say, the discovery of the double helix. And there were two scientists named Watson and Crick, okay, in the 1950s that actually stole her picture almost from her. Basically, they did steal her picture, and they used her information and her pictures to come up with and claim the idea of the double helix. So the structure you know of as DNA is that you can kind of see in the background of this PowerPoint here is actually due to the fact that Rosalind Franklin took these pictures, and she actually died from cancer because of these number of X-rays that she was taking and her work that she was doing, and never fully got credit for this because she couldn't win a Nobel Peace Prize because they don't hand them out to people that had passed away. So she kind of got short end on that one, on the deal. Okay, so moving forward a little bit more with the double helix, and you can see there are two chains that are held together by what we call complementary base pairing. Okay, and basically what happens is you have specific bonding occurs between A's and T's and G's and C's. Okay, so A always goes with T, and G always goes with C. Okay, so you need to remember that. I always remember at. A goes with T, and then G and C kind of like the nutritional store. You know, G and C, the size of cells of vitamins, all that stuff. It kind of helps you remember those. Okay. There's another scientist. His name was Erwin Turgoff, and in his research and going through the double helix and going through DNA and all that stuff, he had found that A always equal T's, and the G's always equal C, meaning the number of A's you have, or the percentage of A's you have, is always equal to the percentage of T's, and the percentage of G's and C's are always equal to each other. So this is referred to as Chargaff's rule, that A's equals T's and G's equals C's, or C equals G, however you like to say it. Okay, this is all due to his research and work that he had done on the experiment of DNA. Okay, so looking at this structure here and what you should get out of this, okay, to this picture that you see here, is that you can see the A is always bonding with T, G always bonding with C, okay, and what they're held together here in the middle by what we refer to as hydrogen bonds, okay, and what you will notice, okay, that the A's are always bonded, are double bonded, and G and C is always triple bonded. Okay, so you should know that between A's and T's there's a double bond. Between G and C there's a triple bond. Okay, and looking at kind of more of the chemistry model of it where you can kind of see this and how it lays out. All right, so you can see here's the triple bond that's occurring between G's and C's, and here's the double bond that occurs between the A's and T's. Okay, so again, through the screencast, the second screencast, you should basically be getting the idea of what DNA is and what the DNA structure looks like. Double-stranded, wound up. It's a double helix. They're built up by these nucleotides. Nucleotides have 3 different parts to them. You're talking about your phosphate, your nitrogen base, and then also your phosphate, your nitrogen base, and then your 5-carbon sugar. Okay, and basically your 5-carbon sugar and your phosphate is the backbone, and the rungs of the ladder, or what's between them, is your nitrogen base, and that's what holds them together. Okay, so this was the screencast for the second segment of the central dogma, where you're looking at the DNA structure and its function as being the genetic material and the transferring the genetic information from parent to offspring. Hello again, biology students. This is Mr. Parker here again, and this is just the end of segment 2. And what I'm doing is going to go over some of the diagrams and notes that you should put on some of your diagrams within your packet for segment 1 and 2 that will help you complete your notes for the DNA structure, genetic material, and function. Okay, so the first one we're going to look at is on page 35 in your packet. As you can see here, we're looking at Griffith's discovery of transformation, and this is when he was working with the mice and the different types of bacteria. Okay, as you can see in this particular his first step, what he looked at here was the S bacteria. Okay, and notice in the S bacteria, it killed the mouse. Okay, so he injected the mouse with the S bacteria, and that bacteria killed the mouse. Okay, and in the second one, he treated with what he referred to as the R bacteria, and it did not kill the mouse. Okay, and then if you look at section 3 here, what he did was he took that S bacteria that, if you look back here, it did kill the mice on step 1. Okay, but what happened was he treated it with heat, and it didn't kill the bacteria, but it did not allow the bacteria to reproduce more bacteria. Okay, and what he had found that, well, the mice, the bacteria can't reproduce more, so the mouse wouldn't die. And then what he did in the third step, okay, he took that S bacteria, okay, that was heat treated, plus the R bacteria, okay, that you can see here, and by doing so, what he had found, what happened was that it killed the mouse. Okay, so this heat-killed S bacteria that did not kill the mouse in step 3, but when he mixed it with R bacteria that was reproducing regularly, what happened was that the DNA was able to transfer its information from the S bacteria to the R bacteria, so it killed the mouse. So again, this was proving the idea of transformation that occurred within the organism. Now, we don't know DNA at this time. They didn't know DNA was actually the transferring material, but he knew that something was transferring the genetic information. Okay, if you flip that over, this is page 36. Again, we're just kind of reinforcing the idea of this is Hershey and Chase. And remember, with Hershey and Chase, they worked with bacteriophages, and these are basically viruses, as you can see here, that will attack bacteria. Okay, and what this did is just show that DNA, again, is that genetic material that's going to transfer the information. All right, so what they originally had done was he took sulfur, okay, and he labeled the protein, which was this outside coat that you see here, and then he took phosphorus and he labeled just the bacteria. And what he did is he allowed the bacteria the virus to infect the bacteria that you can see here. And then what he had noticed was, okay, that virus moved into the cells. Okay, the virus's DNA moved into the cell. And so it was proving here, okay, these coats that were traced with the sulfur remained in the phages and not actually being transferred into the bacteria. So this was proving again that that DNA, for the first time, DNA was the actual genetic material that was transferring it. Okay, so those two experiments. Now let's go ahead and look at a little bit about the structure of DNA. And we had talked about before, the DNA this kind of goes along with segment 2. This is page 45 in your packet. And you can see here DNA, it's got the double helix that you can see. And remember, here you can see that these are the building blocks, right? Your nucleotides are your building blocks of your nucleic acid. It's made up of your phosphates, your sugar, and your nitrogen base. Okay, and you can see here, according to the picture, the diagram, you can see here between the G's and C's are your triple bonds, and between your A's and T's are your double bonds. You can see that going all the way out through this. And you got the double helix that's occurring there through the structure. Okay, and there's also another picture on page 46. Okay, you can see it's kind of illustrating the same thing. And then on page 47, if you could look at that with me on page 47 in your packet, okay, you can see here is our sugar phosphate, okay, and we have a sugar phosphate backbone, and you have your base pairs in here, or also referred to as the rungs of the ladder, all right. And then if we're looking at this structure here, that's referred to as your polynucleotide chain, because what they have done is they've linked the nucleotides together in this chain, creating a longer, what we refer to as the polynucleotide chain. And you can see here between the A's and the T's again, you have the double bond, it's a hydrogen bond occurring, and then you have the G's and C's, which is a triple bond occurring between those. Okay, and you can see here in this little call-out box is your nucleotide structure. All right, and this double helix, if you remember from our notes, okay, Rosalind Franklin was the one that did the x-rays of this and helped Watson and Crick develop the idea of what the double helix actually looks like. Okay, so a little bit more information, and if you flip over to the back side here of this, again, it's showing you the bonds between A's and T's and G's and C's. It's showing you here that you have your guanine and adenine, which we know are your purines, and then you have your pyrimidines, okay, which is going to be your cytosine and thymine. Triple bond here, double bond occurring here. Okay, and the last thing we wanted to go through was the idea of what we call complementary base pairing, and this was in your notes, but let's go through it one more time. And you'll have to be able to do this on one of your quizzes, okay? And basically what complementary base pairing is, if I give you a strand, let's say A, T, G, and C, that's one side of the DNA molecule. Now I need the second side. You would have to be able to come up with, all right, what's the second side? Well, A is always bonded to T, okay? T is always bonded to A, G to C, and C to G. Okay, so you need to be able to do that, and then you should put in the right number of bonds. So A is bonded, remember it has double bonds. Okay, G and C are triple bonds. Same way. Okay, and each bond is represented, or each line equals what we call 2 electrons. Okay, so if I ask you like A double bonded with T, if I ask you how many bonds are there, or how many electrons, you would just say there's 4 electrons. Okay, if I ask you how many between G and C, you would just say there is 6 electrons, because there's 2 electrons for each one of the bonds that are occurring there. Okay, it creates a stable bond. All right, so this was just a little more information to the idea of the double helix, into the DNA structure and function, and hopefully you put a little bit more, a few notes onto your diagrams, and that will help further your information. All right, this was the ending of the segment 2 of the structure and function of the DNA, the screencast that we have done.